Thank you very much, Brian, and thank you for inviting me to talk today. So the theme of my talk is motor neuron disease. Are we really making progress? This was the auditorium at the end of my last talk. So one person asleep, one person needing alcohol, and the rest of the room empty. So hopefully it won't be like that today. 23 years ago, the situation for motor neuron disease was very different from how it is today. There was Bill Clinton, Nelson Mandela, and Tony Blair had just been elected to office, just to cast your mind back into the context of those times. Oasis were in the charts. Pulp Fiction was on at the cinema. The Sony PlayStation was the best kind of gaming machine available. Computers looked like that, and mobile phones looked like bricks. For motor neuron disease itself, we thought that there was one condition, motor neuron disease. There'd been one gene discovered at that time, which was SOD1. Riluzole, which is now a standard treatment, was still in clinical trials. There was no way to measure involvement in the form of, for example, the Ellis-Scorial criteria, which you may have heard of, or function or stage of motor neuron disease. There was no knowledge of the effects on thinking that happens to some people, and there was no real way to treat symptoms related to breathing or swallowing at that time. So the situation now is vastly different. Not one of those things is true. So the short answer to the question, are we making progress, is yes. But the detail is slightly different. This is my head reconstructed in 3D from MRI images. I'm just going to chop it through. And here you can see my brain revealed. This is where some motor neurons live. These are called upper motor neurons. They live in the motor cortex, and they send their wires down into the spinal cord or the brainstem. So the one I've illustrated here goes to the brainstem. And then they talk to another neuron called a lower motor neuron that starts in the brainstem or spinal cord and goes out, in this case to the tongue, but it will go out to a muscle in your arm or your leg as well. If we damage the upper motor neurons, the ones in red, then the symptoms are weakness and stiffness, which is called spasticity, and brisk reflexes. And the brisk reflexes are when the neurologist taps your tendon and the arm jumps or the leg jumps. If you damage the lower motor neuron, then the weakness is still there, but there's wasting this time because those nerves release chemicals to keep the muscles large. There's also fasciculation, which is flickering of the muscles, and there's a loss of the reflexes. So when a neurologist sees this apparently paradoxical combination where you might see wasting with brisk reflexes, which is a combination we should never normally see, then we immediately suspect that motor neuron disease might be responsible. And this is, as we know, one of the manifestations. So we can see here muscle wasting. We can see the hands are flexed, and that's because as the muscles waste, the flexors are stronger than the extensors in the arms, so your fingers will flex because those muscles are always slightly stronger than the ones that straighten the fingers. And on the back of the hand, we can see muscle wasting and guttering. So this is the definition of motor neuron disease. This is taken from the USA, from the National Institutes of Health and the NINDS, which are major US institutions. In ALS, which is what they call motor neuron disease, both the upper motor neurons and the lower motor neurons degenerate and stop sending messages to the muscles. So this is what it looks like. We've got ALS in the middle there, but actually not everybody presents with a mix of upper and lower motor neuron damage. Some people have a pure lower motor neuron problem. They don't really have the brisk reflex and stiffness. And those people, we say, have PMA, progressive muscular atrophy. And other people have only the stiffness. They don't really have the muscle wasting. And those people, we say, have primary lateral sclerosis. And the question is, where is the boundary between ALS, PLS, and PMA? Is there a boundary? Actually, when we look in great detail scientifically, you can see there's no real boundary. These are all really the same condition. There's also another problem. Some people have motor neuron disease confined to the swallowing muscles, and other people have it confined to the arms or legs for a long period of time. So those people have an isolated form of motor neuron disease. Is that the same as ALS, typical ALS? Actually, we think it is. So we have one component, which is a clinical description here, where you've got PMA, ALS, PLS, progressive bulbar palsy, pseudobulbar palsy, lots of different classification systems. but we all lump them together into one thing, which is the diagnosis. So there's a difference between the diagnosis, which is a kind of lump, and the clinical description. So throughout my talk, I'm going to call motor neuron disease ALS, and I mean the diagnosis ALS. And I do that because most neurologists and most scientists think of it as ALS. We don't really think of it as motor neuron disease. But they're actually synonymous, as you can see here. Now, there's other features that we see, clinical features, which we're beginning to recognize. So some people, about 5% of people, have severe problems with thinking ability, and that is called frontotemporal dementia. And up to 30% of people will have some subtle impairment, which you can detect if you do detailed testing. And there's lots of other descriptive terms. You may have heard some of these, flail arm or flail leg syndrome for people with generally floppy weakness that's quite symmetrical in the arms, for example. Some people have ALS beginning extremely young, so we'd call that young onset. Other people in their 90s, that would be old onset. Some people have very aggressive disease. Other people have extremely slowly progressive disease. Some people have onset with a swallowing problem. Other people have onset with a walking or hand problem. So all of these different clinical descriptions are there, and we lump them into one box, but are they really all the same condition? And you could ask, why does that matter? Why on earth would it matter whether they're all the same condition or not? The reason is the theme of my talk, which is precision medicine. When you have a chest infection, we don't just give one antibiotic for all chest infections. We test the type of chest infection you might have. We say, have you got TB? Have you got pneumococcus? Etc. And we treat the specific type of chest infection you've got. Because we've got a deep understanding of the causes of chest infection, we can target that treatment precisely to the type of chest infection, and now pneumonia isn't usually a killer. The same for blood groups. We don't just give any old blood to someone who needs a transfusion, we match it specifically to that person's blood type. So we need to do the same for ALS. Because at the moment, we just treat it all as one condition, we just give one treatment and we say, well, that should work. And maybe it does for a subgroup of people, but we can't tell. So how can we do that? What you can see here is part of the problem. This is the night sky. So I want you to think of that as the patient journey from when they develop their first symptoms all the way through to when they see the doctor. You can see the constellations, you can see the sky is changing and moving all the time. There's some bright objects, you can think of those as symptoms. Sometimes those symptoms are hidden by clouds. Other times you get a very large symptom. You can see here a very bright object about to appear. So when you see somebody and try and classify what type of motor neuron disease they might have, you're just seeing a snapshot of what is actually a continuous moving process. And the timing of when we see the person and the timing of when we classify them will affect what we're going to describe it as. So here's a snapshot from that movie. What can you see on it? Anyone? The moon, yeah. Anything else? It's not a plane, it's a shooting star. Anything else? That's a shooting star, not a comet. So stars, did somebody say? A light on the horizon, yeah, very good. Okay, I want you to focus on this star. Okay, so imagine that's one of the symptoms that the person comes to you with. So this star, if we zoom in on it a bit, can you tell what it is? If I zoom in a bit more, can you tell now what it is? There are some clues if you look very closely. I'll zoom in very deeply. Can you tell what it is now? Yep. It's not Venus, it's Jupiter, but very good guess. It is a planet. Okay, it's Jupiter. You can see 3 of its moons. So we initially just dismissed it as a bright star. It's not a bright star, it's a planet. And that makes a difference as to what you could do with it. It's the same for motor neuron disease. We need to know exactly what the symptoms and signs are and what they mean and how they relate to everything else that's there to be able to tackle it properly. So knowing what to look for is important, and then we can correctly classify people using the correct observations, and then we can hopefully design a treatment that's specific. But as I said, we only see a snapshot of what is a continuous process. So what's the best way to classify ALS? Neurologists classify it one way. We actually gave information from a set of patients we'd seen in the King's Motor Nerve Clinic over a large period of time. We classified all of those people who we saw into different types of ALS as neurologists. We gave that same information to a computer, which could do machine learning and just look at the patterns itself. So as neurologists, when we classify people, if that has some biological meaning, then it might relate to something else biological that we didn't give it. So in this case, what we did was we measured how long did people survive with their motor neuron disease, and we split them into the different types of motor neuron disease that we'd seen. So this is called the Kaplan-Meier curve. It looks complicated, but it's easy to understand. So this is survival in months from onset, and this is the proportion of people left alive at any point. So everybody's alive at the beginning, and then if somebody dies, the proportion of people left drops. And what that means is the further the curve is to the right and the more horizontal the curve is, the longer that group of people has lived. And you can see the curves are separate. People with this flail leg or flail arm syndrome pattern where it's generally floppy, those people do better than people with typical motor neuron disease here. So our classifications as neurologists do make some biological sense. They correspond to something biological that's happening. How did the machines do? They did like this. This is the same information from the same initial visit. So the machine was able, the computer was able to pull out one group of people here who never died over the course of the study, many years. This group of people who had an average survival of 15 years. This group of people with an average survival of 10 years, and then this group of more typical motor neuron disease. So there are some things hidden in the way people present that we're not spotting. We're just saying this is the night sky, but actually there are some Jupiters and other things in there that we need to spot and know what they are. What about the timing? So that night sky movie I showed you, if I could freeze a little bit of it, if I told you this was 1 hour's worth of the movie smeared, then you say, well, that's good. I can tell things are rotating around the night sky. I can tell that's the center up there somewhere. And you can see roughly how far the sky is rotating. The problem is you can't tell where that comet is, the shooting star. You can't tell where the constellations are anymore. You lose a lot of information by doing that. And what would be better would be if we could somehow freeze that information and make the ground move, like this. So now you can see here are the constellations. There's Orion, for example. The ground has moved. But what we've done is we've frozen that 1 hour of time and summarized it in one go. And we call that clinical staging when we do it in diseases. You actually sometimes get more information by doing that. So for example, now you probably can't tell because it's too light in here, but the Milky Way is now visible around here. I can see it on my screen. So staging systems are important. They're an objective indication of disease progression, and they allow you to say, well, I'm going to say this portion is one stage, the next portion is another stage. They let you compact a period of time of that continuous process into one description. They can guide treatment for that reason. In fact, they're used, for example, in cancer all the time. You decide the type of treatment you're going to give to somebody with cancer based on their clinical stage. They can guide health economics because you can say, well, how much does it cost for somebody at this stage of the disease compared to this stage of the disease? And they can also act as an endpoint in clinical trials, which can make the trial shorter because you just have to wait for people to reach a particular clinical stage. And if you can have shorter clinical trials, it means you can do more of them because they're cheaper to do. And therefore, we're more likely to find a treatment more quickly. The system we use is the King stage, which actually is now used internationally. It correlates very well with intuition. So what we did here is we gave little clinical vignettes, little clinical descriptions of people to neurologists, nurses, other healthcare professionals, and we said, do you think this person's at the beginning of their motor neuron disease, at the end, somewhere in the middle? Give it a number between 1 and 4. And then we applied our staging system, and we showed the two match very well. The King stage also will match with the functional rating scale, the ALS functional rating scale, which is a set of questions designed to decide how badly is the motor neuron disease affecting your day-to-day function. And as the disease stage increases, the function drops. And this is a study from South Korea, for example. They looked at the socioeconomic costs, and you can see that as the disease stage increases, the costs go up. It becomes more and more expensive as the disease goes on. And that's not just for the government, it's also for the patient. So we can describe ALS by features at a particular stage, but what about the causes? The problem is that causes and symptoms are intertwined. So at the bottom here, I've got 3 example causes, green, red, and blue. And up here, some of the types of symptoms and signs we might see up at the top. And you can see, for example, this green cause causes problems with thinking, problems with control of emotion, and swallowing difficulty. Whereas this red one causes some difficulty thinking, motor neuron disease, and the blue one just causes typical motor neuron disease with no impairment of thinking. So you've got what's called pleiotropy. So that means one cause results in many different outcomes. But on the other hand, we've got another thing we call heterogeneity, which is where one outcome has multiple causes. And that's important because if we're looking at motor neuron disease and we're just saying, well, it's all one condition, actually we need to know this is one type of motor neuron disease, that's another type, based on the cause of that condition. Because if you're going to make a treatment, it's got to attack the cause, ideally. Causes act through mechanisms, so have we got any clues? There's some unexplained things about motor neuron disease. They're very difficult to explain. Things like there's an increasing risk with age. The older you get, the more likely you are to develop motor neuron disease. Why is that? There doesn't seem to be a single cause. We know genetically there are many causes, but we haven't even been able to find any environmental risk factors yet that are reliable, but there must be some. One gene variant can cause multiple different diseases. You can get, for example, a mutation in a gene called C9orf72. In some people, that causes typical motor neuron disease. In other people, it causes frontotemporal dementia. In some people, it causes both. In other people, it can even cause schizophrenia or Parkinson's disease. How can one gene result in all of these different types of disease? There's also a thing called reduced gene penetrance. That means that even if you carry a disease gene for motor neuron disease, you're not necessarily going to get motor neuron disease. Why is that? Why do some people carrying the disease gene get it and other people don't, even though they're carrying the same variant? Another problem is it seems to start in one area and then spread. Why is that? Why doesn't it just affect the whole body in one go? If you have a rash, it affects the whole body in one go. It doesn't start in one little area and spread usually. Some of them do, but they don't normally. And it also seems to be specific to motor neurons. Not completely. It does sometimes affect other neurons, but really it's quite closely linked to motor neurons. Why is that? So we can again look at the cancer world and see if that can give us any clues. In cancer, we know it's a multi-step process. So your cells have to go through a set of steps. Some of those steps will be things like you happen to have inherited some cancer risk genes. You might smoke, which will make other cells have mutations in them when they're already carrying these cancer risk genes. Then this cell that's got both of those hits, you might get a virus infection. And that virus infection might activate some other process in your body. You then get a bacterial infection, which leads to inflammation. And then maybe you drink coffee or some other thing, like I'm not saying coffee is a risk factor for these, but something like that, coffee is a risk factor for some cancers, that it does something else to those cells that have been through all of those processes, and then the cancer begins. It doesn't begin if you break any of this. So if you don't have this, none of these matter, unless you can cause this by another cause, like here you've got smoking leading to both of these problems. So this is what happens in cancer. Could it be what happens in motor neuron disease? Could ALS be a multi-step process? If it is, then one of the things you know we can show mathematically, I'm happy to share the maths with anybody afterwards if they want, is that it predicts a specific pattern of the relationship between the rate of motor neuron disease you see at any age and the age itself. Which means that if we plot a graph of the age of people and the incidence, the likelihood they're going to get motor neuron disease at that age, it should be a straight line. And for the mathematical reasons that I'm not going to show you here, the slope of that line tells you how many steps there are to develop motor neuron disease. So when we knew this, we looked at a population register we have in England called the Southeast ALS Register. And when we did that, we saw that we did indeed get a straight line. So this number here, r squared, is 0.95. That tells you how likely, how straight are these dots on a straight line. If that number is less than 0.8, then it means they're not on a straight line. So 0.95 is really good. That means that they really do show a straight line relationship, which means that it could be a multi-step process. But we thought, well, maybe that's just a fluke. This is just one register. So we went to our colleagues in the Netherlands. They had this result. We went to our colleagues in Italy. They had this result, in Ireland and in Scotland. And when we put them all together, it was a very nice straight line. And that tells us that the slope of all of them must have been the same, otherwise it would have been wonky. So it does look like it's a multi-step process, and actually all of those have a slope of 5, which tells us that ALS looks like it's a 6-step process. So there's 6 molecular steps you need to develop the disease. And that explains quite a lot of those things that I told you were mysteries. For example, the increasing risk with age comes about because you have to have lived long enough to have gone through all 6 steps. The fact there's no single cause is because lots of things could result in one of the steps. For example, if one of the steps is a particular type of mutation, and we know smoking can cause mutation, well, so can inheriting that mutation, so could radiation from X-rays or traveling in an airplane, etc. So you could have one of the steps caused by lots of different things. Environmental causes would be difficult to find because you might have one pathway for one group of people and a different pathway for other groups of people, and because there are 6 steps to go through, you're going to dilute all of those different environmental effects and make them more difficult to detect. One gene variant could cause different diseases because if you have a gene as step 1, your gene variation, and then you go 1, 2, 3, 4, 5, 6, you get motor neuron disease, but you go 1, 2, 3, 4, 5, 7, you get a different disease. So if the 6th step is a different thing, that would explain why you can get different diseases from the same disease gene. It can start in one region because only those neurons will have gone through all 6 steps, and it would be specific to motor neurons because one of the steps would require something that only motor neurons have. So a multi-step process would explain a lot for motor neuron disease and might be important because you could then target a treatment at any one of the steps you can discover, and that should stop it happening. So are there any causes that we know of? Because I know you're not geneticists, I'm just going to give you a quick genetics lesson. So this is DNA. DNA is a long string, and it's got teeth in it, 4 different types of teeth, just like you've got 4 different teeth. So you've got incisors, canines, premolars, and molars. DNA has G, A, T, C. And it's the order of the teeth on the chain that matters. So the order of the teeth is a computer program that tells your cells how to make more cells and how to make all the different cells of your body. And that DNA is about 1 meter long in every single cell. So it's a huge, long, strangly thing. And if you're old enough to remember telephones which have a cord, you'll remember that they get tangled up very, very easily. So your body stops that happening by wrapping up the DNA in these purple things called histones, those blue wiggly things you can see there. And then it's coiled, and those coils are coiled, and the coiled coils are coiled, and it keeps getting coiled and coiled and coiled until it's packed down into little things in your cell. If this was the size of a ladder, if each of the teeth in that DNA was the size of a ladder, it was stretched to the moon and back twice for every single cell of your body. So it's a huge amount of information packed into there. I'm just going to let this finish because it's almost finished, but these are the final bits of coiling, and you can see that they make things called chromosomes, which are inside the nucleus of each cell. And that DNA has to be copied faultlessly every single time you make a new cell of your body. And when you pass that DNA onto your offspring, onto your children, that DNA replication has to be absolutely perfect. This is the machine that does it in your body. This little thing here is a machine. So this is the DNA coming in. The forward strand just goes straight through and gets copied, but the backward strand, because it's 2 strands wound around each other, has to be looped through this thing and fed backwards and then unlinked. I can look at this lots and lots of times and never quite understand it. But it's doing this a huge number of times. You're making 1 million red cells a second. That's just your red cells. You're making hair cells, skin cells, liver cells, gut cells, etc., all the time. So you're copying all of this DNA millions and millions of times every second. You mustn't make a single mistake. So you mustn't make any errors at all. Otherwise, you've got a mutation. And if you've got a mutation, that might lead to disease. And the challenge for us as neurologists, geneticists, is if we're looking for genetic causes, we've got to search that stepladder that goes from the Earth to the moon and back twice for a single rung, a single step that's the wrong one out of those 4 teeth. So it's quite a challenging thing to do. And to do it, you need huge sample sizes. So we're part of Project Mine, which Brian mentioned earlier, which is an international collaboration. It collects DNA, and we've also been part of this joint program on neurodegeneration research, JPND. I lead the Strength Consortium. There's also other ones, Sophia and ALS Care, that we're involved with. And the important thing about all of these studies is each person who goes in gets a single international ID code, which means we can all share our data. So we're not just collecting information in the UK and that's it, it's a little silo. We're sharing it across the world, and everyone else is sharing their data with us. And that's really important. Everyone's collecting the same information, so my information is compatible with the information collected in America or Holland. Everyone collects it in the same way. which is important. So you don't just get an effect that, oh yes, this is a result here, but actually it's because you collected it this way and they collected it that way. All of the information is securely shared, so we have to follow data protection principles across Europe, and we have to have international legal data sharing agreements, so it's all legally compliant. And doing that, we've made some breakthroughs. So the C9orf72 gene was found this way when we just analyzed 12,000 people for about 1 million genetic variants each. So here you can see a plot. This red line here with this dot is a signal around these 3 genes. We now know that's the important one, the top one. And that's responsible in the UK for about 10% of people who have motor neuron disease, even if they don't have a family history. So it's an important genetic variation. But as I said before, even if you carry that disease gene, it doesn't mean you're going to get the disease. It's a very strong risk factor, though. But that's with 12,000 people. We've just done the study again with 39,000. And now you can see this signal is much, much bigger, but also you get all of these other signals. So we can discover new genes this way. The more samples we get in, the more we can do. This is 17 million genetic variants tested across 40,000 people internationally. Now we're sequencing whole genomes. So 3 billion genetic variants we're looking for per person. To put that in context, Google just recently made an announcement. If you're on an Android, then, or if you use Google Photos app on your iPhone, your pictures will automatically be uploaded to Google's servers as a backup. And Google recently announced that it's got the entire world's video and photo uploads assessed, and it's 13,500 terabytes. That's 13.5 petabytes, if you know the next stage up after terabytes. So they were announcing that because they were so pleased it was such a huge amount of information. But Project Mine, our whole genome sequencing project, will generate 22,500 terabytes. So it's much, much more. You can't just email that to somebody. You can't even open it because you need 22,500 hard drives just to have it before you've even backed it up. So you can't plug them into a computer. It's an incredibly challenging thing to do. And that's because genetic technology is advancing much more rapidly than computing technology. These are the gene discoveries that have been made. So the size of the sphere tells you how much research has been done into it. So you can see there's quite a few big ones here. But what I really want you to notice as well is that the green, I can't remember if I've put this on here. No, I haven't. I'll tell you it. So the green, blue, and red colored labels, those are different themes. They're different types of problem that those genes are involved in. So you can see we're beginning to build up a jigsaw of what might cause motor neuron disease. The other thing you can see is, I've drawn this black line through these dots. Gene discovery is doubling every 4 years, roughly. So we've got 25 genes now. In another 4 years, there'll be 50. In another 4 years, there'll be 100. And that's important because suddenly, we're hoping, suddenly all of the different bits will fit together and we say, oh yes, well this is a pathway. And once we understand that, then we can say, well, then it's obvious how to treat motor neuron disease. You just need to do this, or you just need to avoid that. So that's why it's important to understand these causes. It also allows us potentially to do gene therapy. And that's where if we discover a gene variant in somebody, we could cancel out the effect of the harmful mutation. So in ALS, that would mean switching off the gene. And there are techniques to do that I'll come to in a little bit. What about lifestyle and environment? Is it something that people do that makes them at risk? One of the ways to discover that is to make a national register. And then when you know where everybody lives who's had motor neuron disease, you might see clusters. For example, you might see a cluster, you might see consistently people always live near radioactive areas, or they always live near where pesticides are used, or there are nuclear power stations nearby. You might see some pattern like that. But also, you could ask people lifestyle questionnaires and see if there are differences in the lifestyles of people who develop motor neuron disease from those who don't. So to get a national register, you have to be able to identify every single person who's had motor neuron disease ever in the UK. And that's not a particularly simple thing to do. This is the general pathway of people in the UK. So the GP sees somebody who doesn't yet have a diagnosis. They start to think, well, maybe they do have ALS. They'll refer them to a neurologist, or they'll refer them to someone else who then refers them to a neurologist, or they'll refer them directly to a care centre, a specialist care centre. If they just go to a district general hospital neurologist, that person will then refer to a care centre usually. Not always, but usually. So we can capture people by asking all the neurologists we see, please could you send us the details of everybody who's got motor neuron disease, and also by asking all the care centres to capture that information as well. And ideally, we'd also do that for all of the GPs, but that's a much more challenging thing. because the average GP will see just one person with motor neuron disease in their working lifetime. The average neurologist will see 2 people a year, and our care centre, which is a fairly busy one but isn't that unusual, sees 200 people a year. So the difference in how the effort required to capture a patient is very different. For the National Register, this is one of our planning meetings. So this is Lynn Osher, who's the project manager with Kevin Talbot, who's my counterpart in Oxford. This is my project manager, Anna Kulka, and the computer scientist who's making the register, Sarah Martin. Some of the information you can get from that kind of thing is things like this. This is the lifetime risk of motor neuron disease. So you'll have heard that the incidence of ALS or motor neuron disease is 1 to 2 per 100,000 person years. I don't know if any of you know what a person year is. It's a standard person observed for 1 year. Do you know what a standard person is? I don't. So it's much more meaningful to understand, instead of units of 100,000 person years that don't really mean anything to anybody, what's the chance that a single person will develop motor neuron disease? So your chance is 1 in 300 if you have no family history. Even if you do have a family history but only one person is affected, it's not very different from that. But we can tell that it's 1 in 300 because of these population registers. And that makes it the same lifetime risk as multiple sclerosis in the UK. And multiple sclerosis is something that pharmaceutical companies all want to invest in because they say, oh, there's a lot of multiple sclerosis, we can make a lot of money from it. But MND, they don't see that because people with MND generally don't live long enough to make it a very common disease. But if they did treat it and make it so that it didn't shorten people's lives, they would have a captive audience as big as that of multiple sclerosis. So it's worth it for them if they know this number, 1 in 300. So that's a very useful type of information you can get from a national register. This is all the people using the Southeast ALS register over a 10-year period. This is where they all lived. So you can see some clusters if you look there. I'm going to draw some circles around the clusters I see, which is there. So there's one around this kind of motorway corridor. There's some around the coast. There's this in London. But these can all be easily explained. Well, actually, the motorway corridor one can't, but these can. So the ones at the coast, I told you motor neuron disease, your risk increases with age, and you even saw that with the lifetime risk graph. People tend to retire by the coast, so there'll be more people at risk at the coast. The cluster in London is just because there's more people there, so you're going to have more cases. But even if you take those kinds of factors into account, we can still see that there are some areas that have a higher risk. There's some postcodes with a higher risk, up to double the risk, and other areas where there's a much lower risk. So understanding why there's that discrepancy will be important for finding risk factors. But you can also use it for services. If you found that, well, there's hardly any motor neuron disease care services down this area, but look at all the cases, then you might want to put a care centre there and start telling the NHS, well, we need more support there. So it's got lots of benefits. It's another bit of information that's come out of it. From a lifestyle and exposures questionnaire, 200 cases and 200 controls. They asked lots of questions on education, jobs, addresses, leisure activities, smoking, drinking, etc. The first results that we've analysed show that smoking might be a risk factor, increasing your risk 3 to 4 times. Okay, I'm assuming he doesn't do this regularly. So we've got all of these different ways of approaching motor neuron disease and perhaps classifying it. So the question is, how personalised are the treatments we have so far? Because what we want, as I said at the beginning, is precision medicine. We want medicine that targets each type of motor neuron disease specifically. How close are we to that so far? Well, Riluzole is the main drug we use. It's only licensed officially for ALS. And by that, they mean a mix of upper and lower motor neuron signs. But as I told you, even people who are labeled as having progressive muscular atrophy or primary lateral sclerosis, these other variants, actually do have ALS when you do the detailed scientific studies. So in a sense, it's personalized because they've said, well, it's only for this very large subgroup of motor neuron disease called ALS, but it's not really personalized because actually most neurologists will give it for anybody with motor neuron disease. New Dexter is a drug you may or may not have heard of. It's licensed in Europe and in the UK, but the company hasn't made it available in the UK or in Europe. It's available in the US. You can get it if you contact a charity called ALS Worldwide and pay them, they'll send it to you. There's some evidence that it might improve function, swallowing and speaking function in people who have bulbar involvement. So that's a subgroup where there might be a specific type of treatment that would work. That was a phase 2 study that showed some benefit. A phase 3 study is what we now need to see, is that finding a real finding? And we need to lobby the company to make it actually physically available in the UK. Edaravone, which, as you probably know, is licensed by the FDA in the US, but isn't available in Europe or the UK. That is apparently useful for a very specific subgroup of patients who have early stage disease that isn't particularly aggressive and isn't particularly slowly progressive. That's what they've looked at. It may be that if you look in more detail, you can refine that subgroup down, but it looks like people who have relatively slowly progressive disease early on might benefit from edaravone. So again, a little subgroup of people. There's upcoming approaches that are in trial at the moment, antisense SOD1 RNA. So that's in a phase 2 trial now. The idea of antisense technology is that if you have a genetic variation, so this is what normally happens, you have your DNA, that's transcribed into a substance called RNA, and then the RNA is translated into the protein that makes your body. If this DNA has a mutation in it, then this protein will also have an error in it, and that will cause the disease. So the idea of antisense is you give an antisense RNA that as soon as it sees this messenger RNA, it blocks it, it binds to it and makes it unable to make a protein. So that's the idea of antisense treatment. It's not 100%, but it is targeted at people with these specific types of mutation. So there's already a trial for SOD1, people with SOD1 mutation, which is about 2% of all people with motor neurone disease. There's another study about to start soon, we hope, for people with C9orf72 mutation, which is about 10%. So if those are effective, then we'll be able to say, well, we've got a very effective treatment for this particular subgroup of people. And then it means it's easier to target the people who we don't yet have a treatment for because that's a more homogeneous group. There's also the possibility of deactivation of a thing called HERV-K, which is what's called an endogenous retrovirus. So a retrovirus is a virus, like HIV, that infects people, but has done so in the evolutionary past. And those viruses turn themselves into DNA and then stick themselves in your genome and do nothing. They just sit in the genome and then they're transmitted through the generations. And 1% of your body, 1% of your genetic code codes for your body. 8% of your genetic code codes for these viruses. So 8 times more of your DNA is coding for these hitchhikers than actually for you. We know that in some people, some of these viruses that are usually dormant can activate. Now, in multiple sclerosis, we know that one called HERV-W seems to be something that will predispose you to developing MS. And there's some evidence just emerging that it may be something similar in ALS, that HERV-K activation might be important. We don't know this definitely. It's really very new research. But if that's true, then it's possible that an antiretroviral, like an anti-HIV type treatment, would be useful. Now, these aren't infective viruses, because ALS isn't infective. These are things that are actually transmitted in the genome. As I said, we all carry, 8% of our genome carries these things. So it's not, you're not at risk of infecting people by having this. It is simply that they're activated in some way. So it may be that you could do a blood test and say, well, you've got the signature that shows you've got this activated, so we need to treat it. So that would be, again, something that could be useful. In our initial studies, we showed about 50% of people have this signal. Another approach might be to control the immune system. So the Miracal study has just recruited its first patient, actually. I don't know if you've discussed that already today. You haven't? Okay. So this is a study to use low-dose interleukin-2, which will modify the immune system in people with motor neurone disease. One of the things that's known is that a set of white cells, so white cells are your normal cells that are part of your immune system, they've got a policeman type of white cell called a T regulatory lymphocyte, or a TREG. And those TREGs, they stop the immune system getting out of control. Because your immune system is like soldiers, they want to go around killing all the bacteria. But if you leave them to it, then they'll go crazy and start even attacking your own body. On the other hand, you don't want them to be so calm that when the bacteria invade, they don't do anything. So the Tregs have the balance between activating and calming down your immune system. And there's some evidence that in motor neurone disease, the Tregs aren't working properly, they're underregulated. And low-dose interleukin-2 might be a way to get the levels back up to normal and make them work again. And so that's what this study is trying to do. And if that's successful, then you could do a test and say, well, actually, if we measure your Tregs, we can show that they're a bit low. Let's boost them back up to normal. That might be an effective treatment. So again, you've got a subtype based on cause where you can treat it. So this kind of precision medicine is very important. Future treatments, stem cells offer major challenges. I'm always asked about stem cells in my clinic. The main problem is that the way we would like stem cells to work, which is to rewire the nervous system, at the moment that's just not possible for many, many reasons. The only way we can easily use stem cells with current technology is that they might release something that supports the neighboring cells and stops them from dying. Probably, if that's going to work, the cells will need to be patient-specific, just the same as when we do a transplant of any kind, like a bone marrow transplant. It's the same as giving a blood transfusion. You have to match the donor to the recipient. So those will be very, very personalized treatments. So I hope I've shown you that this is the patient journey. We see a snapshot. We've got clinical staging or the disease trajectory. All of these different ways of describing what's going on are just ways of summarizing what's a continuous process. And it's a very tricky thing to do properly. But by doing that, we can hopefully find subtypes of motor neuron disease and therefore have proper personalized treatments. So over 23 years, I would argue we have made massive progress, both in our knowledge, measurement, and approaches to treatment development. I think precision medicine is probably the way forward. Every single person is different, so every single person needs their own specific treatment. But we have to understand how to distinguish the different types of motor neuron disease, and then we can design treatments that are specific to each person. If you're wondering, this is the symposium when it was in Brussels a few years ago. So I thought there was going to be a thank you slide there, but it seems to have disappeared. But thank you very much anyway.